Good morning, everyone. Hope you're not too tired from the fun evening last night. And today is the second day of the 15th HOPE meeting. First, we have Dr. Paul Nurse's lecture. Let me introduce the moderator for this session, Dr. Takashi Toda from Hiroshima University. Please welcome Dr. Toda.
Good morning. It is my privilege to introduce Dr. Paul Nurse this morning. Dr. Nurse is currently the director of the Francis Crick Institute in London, which is the largest research medical institute in Europe. He is a geneticist and a cell biologist and has been working on the biggest problem in life science, that is the mechanism on how cell divides. He beautifully solved this long-standing mystery, the story of which you will learn in a minute. Because of this achievement, Dr. Nurse was awarded the Nobel Prize for Physiology of Medicine in 2001 and then received third. He was also awarded the Order of Merit from late Queen Elizabeth II in 2022, just before she passed away. So I heard that the very last task, which Queen Elizabeth II did, is give this award to Paul, Dr. Nurse. And Dr. Nurse is also has many awards and prestigious prizes, which I can't list everything here. But for Japan, he received the Order of the Rising Sun in 2018 from Emperor Naruhito. His seminal work of the cell cycle has changed our view on how cell divides. And also, this discovery paved the way to the development of many drugs against human diseases, including cancer. He is also actively involved in policy making by government or politicians, and also public understanding of science. And recently, 2021, I think, he wrote a book entitled What is Life? That book is now translated in 22 countries, and including Japanese, and I read it both in English and Japanese, and it is really fascinating, inspiring. Lastly, let me briefly introduce my own story as to how I got to know Dr. Nurse. When I was a graduate school student, a very long time ago, the very first paper which I read is Dr. Nurse's paper published in 1976. Many of you didn't exist yet, I think. And I read it, and I found it really fascinating, and in that sense, that changed my life. And later on, from 1995 to 2015, I was working in the Francis Crick Institute, where Dr. Nurse is the director, and our labs next to each other. So this is really also, I was very fortunate to witness the very moment when Dr. Nurse got a phone call from Sweden, which tells the award of to his Nobel Prize. So now it's Paul's turn. So please welcome Dr. Paul Nurse. Thank you.
Good morning, everybody. It's a pleasure to be speaking to you. I must apologize for my voice. I'm recovering from a cold, and so maybe a bit hoarse, so do forgive me for that. I want to thank JSPS and the HOPE meeting for asking me to come. It's always a pleasure, of course, to come to Japan, always a pleasure to come to Kyoto, and a pleasure to speak to you all this morning. I'm going to talk about this topic here. What controls the reproduction of cells? And my talk will be in 3 parts. There will be a little bit of history, quite ancient history, a second part, which was a brief description of the work that did lead to the Nobel Prize, and a third part, which is some more current research. I did this because I recognize some of you are biologists, some of you are physicists, some of you are chemists. So I'm quite happy if you want to go to sleep at different parts, if it's either too simple or too complicated. It's designed in my talk to allow you to actually do that. Now, I want to start actually by asking or telling you, why did I start working on this topic? And I have to say, the main reason was because my PhD was exceedingly boring. Now, all PhD students think their PhDs are a bit dull, but I can tell you mine was really dull. And what I was doing was analyzing amino acids in fungi. For no particular purpose, it has to be said, but that's what I was doing. And I had to run a machine called an amino acid analyzer, just invented by Beckman. And we had a prototype in my university, which is in Norwich, England, which is indeed, David, under Scotland. It's always under Scotland. And this analyzer was a prototype. And what you need to know about prototypes is they basically don't work. That's why they're called prototypes. And in this case, it had a lot of plumbing and columns and safety devices measuring the pressure of the liquids going through the different parts of the machine. The problem was that if it exceeded a certain pressure, it switched the machine off. And then there was a brilliant design feature that when it switched the machine off, it dumped all the data it had collected up to that point. So you could sit with it for 2 hours and 30 minutes, and if it switched off in the last 10 minutes, you lost all the data. My solution to this was to go around the machine with little pieces of paper, little pieces of plasticine, and inactivate all the safety devices. This meant it worked, but also meant it was now really dangerous because it really could blow up. So the only way I could operate it as a graduate student was to sit in front of it and look at the pressure dial and switch it off if it exceeded what I thought was an unsafe level, which was double what Beckman thought was an unsafe level. Why do I tell you this story? Well, one, to give you encouragement if you're a PhD student, we all suffer, okay? But mainly, this meant I thought for many hours watching this machine, what shall I do next? Because this is a serious business, it's a lot of work, so it better be important. So I decided that I wanted to work on something that was important universally for life, and I thought cells are important, because they're the basic building block of all life. I'll say a word about that in a moment. And the reproduction of cells, of course, is the simplest reproduction process and all living organisms undergo. So let's start with a bit of history. This is the first picture of a cell, drawn in 1665 in the city where I live, Oxford, and it's a wonderful picture of a slice of a plant showing the first cells ever seen under this simple microscope. And it was concluded that these cells were the basic unit, structural unit of life. Then, later, Rudolf Virchow, 19th century, had this great quote there: "Every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life." That meant cells weren't simply the basic structural unit, they were the basic physiological and functional unit. And then the second quote in Latin, omnis cellula acellular, all cells come from cells. And this is a dividing fibroblast cell. And since I started talking to you, millions of those divisions have been going on in your body, even in the couple of minutes I've been speaking to you. So this process of division is important. It's the basis of all growth and reproduction of all living things. It links DNA replication to cells and leads to the inheritance of the genome, and it's important for cancer. This is development. We all came from a single cell, undergoes repeated divisions, eventually forms an embryo. And maybe I should remind all of you, every one of you in this room once looked like this. We all were once a single cell. And if you're not interested now, there's a couple of doors at the back, actually, if you would like to leave. We were all once a single cell. Well, it's very important for genome stability, because every time a cell reproduces, a process we call the cell cycle, the chromosomes replicate, that's the DNA, and then those replicated chromosomes segregate during mitosis, M phase shown here. So we have an S phase, an M phase, a G1, and a G2 in between. So the cell cycle, which is the basis of cell reproduction, is also the basis of heredity. So it's important. And when it goes wrong, as I've drawn here, cell reproduction, when it goes out of control, causes cancer. So this is why it was an interesting problem, and this is what I dreamt of while sitting in front of my amino acid analyzer, waiting for it to blow up. So how do you work out the control when you know nothing about a living process? How do you approach it? And nothing was known about it when I started. How can you investigate it? Well, you can use old-fashioned genetics, looking for mutants which are altered in the process that you're interested in. And I chose a very simple organism, yeast, and I did that because whilst watching my amino acid analyzer, I read some papers from eventually a friend of mine called Lee Hartwell, who got the Nobel Prize at the same time, who had started analyzing cell cycle mutants in the budding yeast. I chose fission yeast. That's the one shown on the left. The one on the right is actually the oldest picture I could find of a fission yeast from a Copenhagen brewery to indicate how brewing could go wrong if you had this particular yeast within it. So you could use genetics in a simple organism like this, and I made the optimistic assumption that maybe studying yeast would be relevant to everything else. I will return to that a little later. So the genetic approach. Here we see a fission yeast. We treat it with a chemical mutagen. It will damage some of the genes that are there as a consequence of the chemical mutagenesis. The cells can normally divide, you can see at the top there, but if you were to hit a gene that is necessary for the cell reproduction, then the cells will be unable to divide, but they could continue to grow, because it's obviously trivial if you block protein synthesis or RNA synthesis and so on. That's not closely related to the cell cycle. So what I simply looked for were mutants that could continue to grow but couldn't complete the cell cycle. And they produce these long, elongated cells. Had to use temperature-sensitive mutants, because obviously, if they never divide, they don't survive. So I used a low temperature when the gene would work, and a high temperature where they wouldn't. Now, this is a scheme of what I just described to you. In the middle there, you see the normal cell cycle. And if we have a cell cycle block, CDC stands for cell division cycle, you get these highly elongated cells. And here's some actual examples of it. In A, you see normal cells dividing, and in B and D, you see cells getting highly elongated. And this may have been the paper that Takashi read. It was 1976. This was a graduate student, Kim Naismith, ended up being professor of biochemistry in Oxford later, who was involved in this work, worked with me as a student. What was interesting here, and I mention it, is my supervisor, Murdoch Mitchison. I should point out for David's information, all of this was done in Edinburgh, in Scotland, where I moved. So you've had 2 Nobel Prizes from Scotland in 2 days. And my supervisor was Murdoch Mitchison, and he never put his name on any of the papers I wrote because he said he didn't contribute with his own hands, the old-fashioned way. I published 15 papers in 7 or 8 years there, and he never put his name on any of the papers. I mean, of course, you could say he didn't like my papers, but that wasn't actually true. He was just being completely generous. Now, the point about these sorts of mutants is they define genes that are necessary for proper cell reproduction. But if you remember my first slide, I was actually interested in what controls that process, not simply what was necessary for the process, what controlled it. But I couldn't think of a way of actually getting to it. And then one day when I was looking for mutants looking like B and C, I saw something that looked like B over here, which is dividing at a small size. I called this mutant a wee mutant because I isolated it in Scotland, and wee is the parochial name for small. And it is actually this mutant that unlocked cell cycle control and named after a Scottish word, wee. Now, why was this important? I'd like to say that I thought this would be important and looked for it. It's not true. I found it by accident. I just looked where nature showed me. But if you look back to this slide, what it indicates is that this gene is probably involved in a rate-limiting step overall for the cell cycle. Because if you finish all the processes needed for the cell cycle more rapidly, you will divide before you grow to the normal size. Very simple. I just hadn't thought of it until I saw it. And so this means cells are advanced through the cell cycle because they undergo the reproductive processes more rapidly than normal. Well, I won't go through the genetic analysis, but it took several years and identified 2 genes, one called WEE1, which is in fact the picture I showed you, and another one called CDC2, which could be mutated in 2 different ways. It could be mutated in a way where if you made it completely defective, the cells couldn't divide. But if you mutated it in a more subtle way, so you made it more active, it would divide faster than normal. Turns out WEE1 regulates CDC2. And this was critical because CDC2 turns out to control not only the onset of mitosis, that is when chromosomes segregate, but also controls the onset of S phase when chromosomes are replicated. 2 very different processes, but both controlled by the same gene. I thought you might be interested to see what I looked like at this time. Can you recognize me there? I'm the one on the left, okay? It's when I had colored hair, okay? I wasn't always white. It was also quite long too, as you can see. Actually, I still have hair, so that's a good thing. So that's what I looked like. And this is the model I came up with when I left Scotland to go back to England to carry on my work, which was this gene, CDC2, was required at the onset of S phase, before cells could replicate their DNA, and also the onset of mitosis. Now, up until this point, this analysis is entirely abstract. I just talk about genes. I have no idea what they do. So it's abstracted from chemistry, biochemistry, from physics. It's just conceptual. So the question is, how can you turn a conceptual analysis into a mechanistic explanation? And the only way you could do that was to clone genes. Now, many of you in the audience who work in biology or medicine will be cloning genes, but at this point, which is 1980, cloning had only been invented about 1 or 2 years before. So I then turned all my attentions to making fission yeast a good organism for doing molecular genetics. And that meant making gene libraries, and this is a picture I drew in 1980, so I just rescued it from there. And I could make a gene library, and did, of the fission yeast gene. And this shows you how, this cartoon shows how it works. On the left, we have a fission yeast cell, which is defective in CDC2, so the cells cannot divide. I sprinkle the gene library on these cells, transforming them, because some cells will take up 1 or 2 of the plasmids of the gene library. And if it takes up a plasmid which contains the CDC2 gene, it will rescue, or potentially could rescue, that function. So these cells, and you see those little red dots, are now making good CDC2 protein and can now grow and divide on the plate. Again, very simple. And then we can extract that plasmid back again, and we now have the gene that encodes the CDC2 the CDC2 gene. And that's the start of molecular analysis. But to go further forward, you had to sequence the gene. Now we could do this, we could sequence that gene in about a minute. But it took us, I think, 3 months to do it using old-fashioned Sanger sequencing. 3 months to actually sequence the gene. Well, when I got the gene, the first thing you would do is compare it to other genes that have been sequenced. But at that time, there were only 50 eukaryotic genes that had been sequenced, only 50. And when I compared it, it just matched a little bit with a viral oncogene, which caused cells to divide out of control, in fact. But biochemically, that was thought to be a protein kinase. What is a protein kinase? Well, it's shown here. A protein kinase adds phosphates to other proteins. Adding a phosphate, which is a large negative charge, to a protein changes the structure of the protein. It can switch it on, it can switch it off. So protein kinases act in many functions in cells as a switch. We now know, because we've done it, we now know that in fission yeast, it phosphorylates 350, if not more, proteins in fission yeast, which are phosphorylated at different stages of the cell cycle. So it has a massive influence on the behavior of the cell. And you see on the right, the protein kinase is phosphorylating 5 substrates which activate processes needed for S-phase mitosis and also for blocking mitosis. So we now had a model that the cell cycle in fission yeast was controlled by a protein kinase phosphorylating proteins required for S-phase and mitosis. But bluntly, who cares about yeast? As I found when I looked for a job and failed to get a job, because nobody cared very much about yeast unless you're into brewing and sake. So I thought, I have to try and find if the same gene exists in human beings, then people would be interested. This was, of course, 15 years before the human genome was sequenced, so I couldn't just go and look at the sequence. But what we decided to do, and we was a postdoc in my lab, Melanie Lee, this is the lady here, was to do exactly the same experiment as we did to clone the fission yeast gene, but this time we used a human gene library. In fact, the first human gene library that was made, it was made by Paul Berg on the west coast of the US. He generously gave it to us only 1 or 2 months after he had made it. We did the same experiment, sprinkled, more or less metaphorically, sprinkled the DNA onto the fission yeast cells to see if humans had a gene that would do the same job as the yeast gene. Now, this was a ridiculously bold experiment because it made the assumption that humans and yeast control their cell cycles in exactly the same way. In fact, in such the same way, you could replace one gene with the other. Even though the last time humans and yeast shared a common ancestor is probably 1,500 million years ago. So 1,500 million years of evolution, totaling 3,000, had gone on between yeast and humans. But we were desperate because every other way, conventional way, we found didn't work. And this, in fact, did work. And when we cloned the gene out, and this was done by Melanie Lee, who later went into biotech, and when we sequenced it, here you can see I compare the human fission yeast and budding yeast gene, which we also showed was similar to this, and you can see there's remarkable identity. Wherever there's identity in the amino acids, I put a box around them. So this meant that humans controlled their cell cycle in the same way as yeast cells control their cell cycle, which, of course, was a surprise. And if humans and yeast do it the same way, every living thing you can see, plants, animals, and fungi, are likely to do it the same way. And this gene can be found in all those organisms. That led to the Nobel Prize. But I'm just going to finish with a couple of experiments that is going on in my lab at the moment. Oh, by the way, I took this terrible photograph. This is the first fission yeast cells being kept alive by a human gene. I took it 2 days after we introduced the gene. And you see, when the plasmid was lost, the cells just produced these highly elongated cells. But when it had the human gene there, it sort of worked. Okay, so this is the Francis Crick Institute, which you've heard I run, I set up. And that's the most exciting slide I have, by the way. Did you like that? It's a dynamic logo. I said when we choose a logo, I want it to look nothing like any university logo. And so we came up with this. And this is the Institute next to St. Pancras Station. And I'm going to quickly show give you some evidence for a different view of how the cell cycle is controlled from what people think from textbooks based on analyzing these CDC genes. And by the way, I forgot to tell you, this kinase is called the cyclin-dependent kinase, CDK, and was worked on by Tim Hunt, who's given a talk here at the HOPE meeting a couple of years ago, who worked on Xenopus, Lee Hartwell in budding yeast, and myself in fission yeast. Now, this is the textbook view. In all eukaryotes, there are multiple CDKs with different cyclins, and in some cases, different protein kinases. And the idea is that the different CDKs come up at different times in the cell cycle, in G1, S, G2, and mitosis. And they have different substrate specificities, which lead to the onset, progression through G1, onset of S phase, progression through G2, onset of mitosis and cell division. This is thought to be regulated in a complicated way. I just want you to look around that and say, oh, isn't it complicated? Okay, don't read it, it's complicated. And then the modelers will get at it and they'll produce something like this. Now, I've deliberately made this small so you can't read it, because I don't want you to read it. I just want you to look at it and feel mildly depressed. Because we produce stuff like this and people think it explains something. And what it is actually like the Tokyo Metro map. And we understand nothing. And since I was one of the originators of this, I felt responsible for this depression. So I thought what we should do in the last part of my working life is to see whether we can simplify this and to see what is the minimal requirement for cell cycle progression controlled by CDK, rather than every little fiddly bit and dumping it in. And I used fission yeast, of course, where we have 4 mitotic CDKs, one CDC2, which encodes CDK1, 4 cyclins, which activate it, PUC1, SIG1, SIG2, and CDC13. And these are thought to have roles, as indeed they do, at different stages of the cell cycle. Then a postdoc in my lab, Damien Kadrusa, liked my idea of trying to simplify things. So the starting point was this. Let's just fuse the cyclin with the CDK, so it's not 2 proteins, but 1, because that would get rid of the controls needed to put them together and to make a stable complex. So he did that, and that worked. This is the fusion. You can see it is a higher protein. On the left is wild type. In the middle is cells containing this fusion, but now deleted for CDC2 and CDC13, which is the cyclin. But then Damien, encouraged by this, went through the genome, and this is the beauty of organisms like fission yeast, and deleted all the other cyclins, SIG1, SIG2, and PUC1, and look what happened. The cell didn't care. It could do everything with just 1 cyclin-dependent kinase. Now, some of you have done biology and will have learned about CDKs in the cell cycle. This is different from the textbooks, isn't it? Completely different. Where the conventional picture is you need different CDKs. This shows you don't. You can actually run it with 1. Encouraged by that, we then looked at meiosis, which is, of course, the reductive division that occurs during germ cell formation, where there's 2 more cyclins, REM1 and CRS1. And Pilar Espiano in my lab deleted those genes as well, and the cells can undergo meiosis with 1 CDK. How can you explain that? Well, the only explanation we could come up with is this one here, which is, it isn't actually lots of different CDKs that are important. It is the total CDK activity in the cell that is the main driver. And that it's a very simple model. Like a rheostat, it starts off at a low level and increases as you go through the cell cycle. And at low level, it undergoes S phase. At high level, it undergoes mitosis. And as it happened, we showed also, my lab showed earlier, that intermediate levels of CDK would block a further round of DNA replication, because that means you only have 1 S phase every cell cycle. Now, we could test this model by mutating the CDC2 gene, CDK1, so it's now sensitive to an inhibitor. This is an approach invented by Shokat, also working on the West Coast, where you basically make the ATP binding site of the kinase larger so that a bulky ATP analog can fit in and inhibit only that kinase and no other kinases. But way we use it was to make the assumption that if you add only a little bit of the inhibitor, you will only inhibit a small amount of the total activity. But if you add a lot, you will inhibit more. So we could mimic the increase that goes through the cell cycle by just adding small, different amounts of the inhibitor. And this experiment is shown here. We got cells just before mitosis. We added DMSO, which is just the solvent, increasing levels up to 300 nanomolar of inhibitor, and this is the percentage of cells in mitosis. And you can see that the black line is actually DMSO, the control, and as you add more and more inhibitor, you eventually block mitosis at what would be, say, between 0.1 and 0.3 micromolar of inhibitor. We then looked at S phase, and I won't explain all of this in detail, and asked the question, how much was needed to block DNA replication? So they blocked in an unreplicated state called 1C here, and what I want you to look at is the bottom right, 5 micromolar delays S phase by about 90 minutes, 10 micromolar, I haven't got it here, by about 150 minutes. So you need 10 micromolar to block S phase and only 0.1 to 0.3 micromolar to block mitosis, indicating you need higher levels of activity at mitosis. This is the fun experiment, I have to say, because here what we did is we blocked cells in G2 with a low amount of the inhibitor. Then we pulsed a high amount of the inhibitor, forced the cells back into G1, then washed it out, and instead of undergoing mitosis, they underwent S phase. So what we're doing here is manipulating where you are in the cell cycle simply by changing the level of activity of cyclin-dependent kinase, the master regulator of the cell cycle. This all works through the protein kinase phosphorylating some substrates at low level and some substrates at high level. But I'm not going to go through those experiments, which are shown here, but this is the summary of what we would say is going on to the right here, my graduate students down the bottom here. At low CDK activity, you get G1S. At high, you get G2M. In the middle, you block. Now, there was one, I want to finish with one more experiment because there was one problem with this model. And that is, if it's true, then the other cyclin-dependent kinases, SIG1, SIG2, and PUC1, should have similar protein kinase substrate specificities. And we couldn't get that experiment to work. We tried to get a SIG2 CDK to actually work, and it didn't work. And then my graduate student here, Saz, devised a system here, and this is a little bit more specialist, and I apologize for you over this, but it's such a nice experiment I wanted to tell you. He took cells which are temperature sensitive for CDC2, M26, so you can switch off the CDK. Then he expressed different fusion proteins, cyclin CDK, including CDC2, CDC13, which works, SIG2 and CDC2, which didn't work. That's shown on the next slide. And you see here that in B, the dark blue or black is the CDC13, CDK1, which works. SIG2, CDK1 doesn't undergo mitosis. Do you see that in B? But what Saz did was then to look at all the inhibitors of CDK and delete them, and found one of them, a phosphatase called PP1. And when he deleted it, you could see cells that could actually go into mitosis. So it was only in the presence of that phosphatase that SIG2 couldn't work. And he then did another experiment which changed the localization of that phosphatase, and you see he got complete rescue. So SIG2 does work, just like CDC13. So the G1S CDK can bring about S phase and mitosis, and the G2M, which is CDC13, CDC2, can bring about G1S and also mitosis as well. Finally, he measured at about 250 substrates using a mass spec, which we've done in the last year or two, and measured the substrate specificity of these two kinases. And over 90% of the substrates have absolutely identical substrate specificity. So our conclusion is that the eukaryotic cell cycle is basically very simply regulated by cyclin-dependent kinase activity that increases like a rheostat and drives S phase and drives mitosis at different levels. When we come to making a multicellular organism like a fly or a human being, we have extra controls on top, which almost certainly require other CDKs. But it's a consequence of making an organism, because if we delete them in a fibroblast cell, that fibroblast cell can divide, but you can't make a mouse, which means it's got extra levels of regulation. So this is the model we are working on now. These are the problems I'm wanting to solve before my hair goes even whiter. What controls this vast range of CDK activity from 2 or 3% to 100%? How do you measure the size of a cell and the ploidy of the cell, which is important, the thresholds and dynamic regulatory behavior, all going on in my lab, which is mainly graduate students. I have to say I've turned it over to essentially graduate students. And that is the end of my talk and the end of the logo. Thank you very much for listening.
Thank you very much. So now, for you to ask question to Paul, raise your hand or go to the microphone. I think 2 stands, and introduce yourself and ask any question, and Paul can answer anything.
Thank you for lecture. So my question is, so if we prepare ingredients of a cell, can we make the cell and cell cycle organism from chemicals or not cell?
So is your question, can we manipulate all of this with chemicals as well as genetics? Yeah? Well, genetics, you probably could, but it's a lot more work, because with genetics we can use the same technique with each gene and control it in particular ways. We've, of course, combined genetics with chemicals, with the SHOCAT inhibitor, which is even more powerful, I would say. So if you want to, of course, intervene in a pharmaceutical sort of way, then of course you need to have inhibitors and activators. And many have been now tested to see if it has effects on cancer, and some of them are successful there. But I don't think this is the answer to cancer, because every cell has to do this mechanism, and so it isn't specific enough. So I don't think those sorts of chemicals will be very useful. Not sure I answered your question, but I hope I did.
Yeah, thank you.
This is Zhou Yang, a physician from Beijing, China, and thank you very much for the presentation. And you mentioned you want to simplify the cell cycle, like only one CDK is enough.
Correct, yeah.
But my question is, what is the philosophy of the cell to have so many CDKs if only one of them is enough? Is that what makes us different from the East?
It's a good question. I think nature wants to confuse us, and it's done quite a good job, to be quite honest. If you think about it, the very first eukaryote could have only had one CDK. Therefore, the prototype we are reconstructing now, 1.5 billion years, 2 billion years later, is we can build it on a single CDK. that will have duplicated over the years. And it is possible that it fine-tunes processes, which we can't detect here because they're only having very small effects, but they have a small selective advantage. It is also very likely that when we make a multicellular organism, as I said, that there will be more specialized regulation going on of the cell cycle, which we need to understand. But I want to stress that when people working with mammalian cells, with human cells, for example, they've been surprised how often they can get rid of cyclin-dependent kinases and cyclins, and it still works. And they tend to say words like, well, the system is very plastic. Whenever you read the word plastic, what is really going on is you don't understand what's going on, so you just say it's plastic. But what actually is going on, I think, is that the level of activity is by far the most important feature. So why so many? It could be that it has a selective advantage, which is small, or it could, of course, have a selective advantage in certain ecological conditions that we do not study in the laboratory. Now, I don't know of any conditions like that. I mean, we've looked at poor nutrients and so on, and there seems to be no impact. But I think that is a possibility. And the third possibility is maybe there's something we really don't quite understand about evolution, that things just carry on there, and if they're not doing much harm, they're not eliminated. And we may need to have a more sophisticated way of thinking about it. So I can't tell you where that comes out. I quite like the last one because that would be a challenge to the, I have no solution, but I think that's worth thinking about.
Thank you.
Thank you.
Hi.
Hi.
Dr. Nurse. I'm Meng Lan from Singapore. I have two questions. Is it okay?
One at a time, please. Yeah.
Okay, I will ask my first one. So what do you think is the role of lysosome in controlling the cell cycle, especially since, mTOR is like important for nutrient sensing that was well studied in mammalian cells and is also conserved in yeast. So like, what's your take on that? And the second question is regarding to the genes that exist in yeast but does not have a functional or sequence homolog in the mammalian cell. How can we study them, and can we learn anything from the model organism like a yeast?
Yeah. Well, let's deal with the second first, because what I think is working with mammalian cells is really difficult, and we sometimes underestimate it. There's good reasons for working with yeast because you can do very precise experiments. We can sequence every strain if we need to check we've just mutated the one gene we think we have. And when you're trying to work out complicated phenomena, when you have many genes, some of which are important, some of which are not, when there's overlap in functions, it becomes very, very difficult to work out principles. And so that's why I think working on simple systems, which you can then simplify further, is important for revealing principles. Then you can see whether those principles apply to more complicated cells. But if you try and work it out with complicated cells, it will be, I think, very difficult to do. So that's my starting point as to why it's useful to do it with yeast in the simple systems, and then guide work that could be done in mammalian cells. I mean, the cell cycle control was discovered in yeast and then found to apply to mammalian cells. And I think that the simplifications we've described are useful. But of course, those complexities, as I explained to an earlier question, do mean something. It's just that you have to take a step at a time to try and work out what the fundamental principles are and then how they are modified. Otherwise, you end up with that metro map and you understand nothing. And it's just not a good way to do science. The lysozyme, the thing about all sorts of things, mitochondria, lysozyme, Golgi and so on, they all have some impact on the life of the cell. They will all have some impact on this. The question is, how central is it? And how peripheral is it? And that will depend on exactly the problem you're studying and exactly the circumstances under which you're looking at it. So once again, I return to trying to keep it simple to get the principles, and then you can add on the complexities. But if you start with the complexities, you will never get the simplicities, and so you will get lost in the metro map.
Okay, thank you.
Thank you.
Hello, Professor. So my question is regarding how much feasible will it be to therapeutically regulate the level of CDKs in order to defer the signs of aging? Can it be a better therapy, or again there are anti-ROS therapies or some other therapies are also there? So
I think you're asking about what is the overall control.
Level of CDKs of the cell cycle controlled proteins therapeutically, if you can modulate. So can we defer the signs of aging in that way?
Right, I'm not sure I've quite got the question, but I think that you're getting at the fact that we're looking here at the core machinery, and there's many other things that impact, as we just said. And a critical one, which I think you were probably referring to, is the growth of the cell.
Exactly. If you can
The growth of the cell. Now, one thing I didn't emphasize here is the cells know how big they are. They can measure how big they are. We do not know how that works. I've been thinking about this since I first saw the Wee mutant in 1975. I still don't know how it works. But it's conceptually straightforward. The cell knows how big it is, and when it gets to mitosis, it knows it's at the size it should do mitosis, and it activates the protein kinase to a high level. We know something about that, but we don't yet know how it monitors its size, which I think is a critical thing. But not only does it monitor its size, it also monitors its ploidy. Because if you take a haploid, a diploid, a tetraploid cell, they go up 1, 2, 4. So the size at which you undergo mitosis is determined by a mixture of measuring your size and the ploidy of the cell. Now, we have recent results, which we haven't submitted yet, where we think that the size measurement is going on inside the nucleus, where, of course, the chromosomes are, partly so they can monitor ploidy. But these are really important problems you're identifying that I'm desperate to know the answers for.
Thank you. Thank you.
Okay. Are you okay? Then, thank you very much.
Thank you. Thank you.
Thank you very much, Dr. Nurse and Dr. Toda. Let's show our appreciation with a round of applause.